Good day to everyone and welcome to our lecture on the cell. I am Nicolay Lumawan Martin, a faculty of Medical Technology Medical Laboratory Science at the University of St. Louis. And the topic cell is one of the first topics in human histology, in which in human histology, this is a course that would deal with the study of the fundamentals of cells, tissues, and organs with a great emphasis on microscopic structure, characteristics, and functions. When we talk about cells, as we know, as a basic definition, cells are considered as the basic structural and functional units of all multicellular organisms. Specific functions are identified with specific structural components and domains within the cell. As we know, the cell would contain different types of organelles, and these organelles would have specialized functions. The cells also can be divided into two major compartments, which is your cytoplasm and your nucleus. Your cytoplasm is the area where most of the cellular processes would occur. It contains or it houses the different organelles such as your mitochondria, your ribosomes, your endoplasmic reticulum, and all other types of organelles. Your nucleus is considered as the brain or the control center of the cell because it houses the genetic information or the DNA, and it actually coordinates activities such as growth, metabolism, and reproduction. We have two basic types of cell in which we have your eukaryotic cell and your prokaryotic cells. Your eukaryotic cells possess a well-defined membrane-bound nucleus that is or that encloses or that houses the cell's genetic information or the DNA. Aside from that, the eukaryotic cell is notably uh, have a feature or it contains rather various membrane-limited organelles. And again, each membrane-bound organelles would have specialized functions. Your prokaryotic cells, on the other hand, lack a true nucleus. It has no nuclear membrane. Thus, the genetic material is located only on a nucleoid region and it's mixed with the rest of the cytoplasm. Also, it does not possess membrane-bound organelles. Therefore, all cellular processes occur within the cytoplasm. Here is an example of a unicellular organism, which is a bacteria. The bacteria now have a cell wall that provides rigidity and shape. It also have a cell membrane that encloses the cytoplasm, a nucleoid region where the DNA is located, again because of the fact that unicellular organisms does not contain a nucleus or a nuclear membrane that would house genetic information. And within the cytoplasm, you can also see ribosomes, which is important in protein synthesis. And some bacteria would also have additional structures such as your flagella or pili or pilus that is important for locomotion and attachment. On the contrary, we have your multicellular animal cells, which are part of a more complex organism. They feature a cell membrane as the outer boundary and a distinct membrane-bound nucleus, which houses the cell's DNA or the genetic material. The cytoplasm also contains various membrane-bound organelles, which includes, for example, your mitochondria, which is important for energy production. You also have your endoplasmic reticulum for protein and lipid synthesis. Your Golgi apparatus or your Golgi body, for example, as well, it's used for protein modification and packaging. Additionally, we also have different organelles such as your lysosomes, which are present during waste breakdown. And you also have your cytoskeleton, which provides your structural support and shape of the cell. If we try to look into the points of differentiation of prokaryotes and eukaryotes, we could see that the DNA of a prokaryote is not again enclosed in a membrane, and usually it's a single circularly arranged chromosome. Your DNA, on the other hand, in eukaryotes is found or it's enclosed in a nucleus. Therefore it is separated from the cytoplasm with a nuclear membrane. The DNA in prokaryotes is not associated with histones, while on the eukaryotes, it's associated with chromosomal proteins, histones, and with non-histones. In biology, when we talk about histones, histones are highly alkaline proteins that are found in eukaryotic cell nuclei. They package and order the DNA into structural units which we call as your nucleosomes. There are five major families, in fact, of your histones. We have your H1 or H5, we have your H2A, H2B, H3, and H4. Your histones H2A, H2B, H3, and H4 are known as your core histones. What is the function of core histones? Your core histones are the central proteins that are around which DNA winds to form the nucleosome core. That's why it's called as core proteins. Aside from that, we have not mentioned H1 and H5 because H1 or H5 are known as the linker histones. Therefore, linker histones, these are proteins that would bind to the DNA between the nucleosomes. They help in stabilizing the higher order structure of chromatin. And again, it's notable to mention in prokaryotes that they lack membrane enclosed organelles, while on the other hand, in eukaryotes, they have a number of membrane enclosed organelles. For eukaryotes, the cell walls, when present rather, are usually chemically simple, unlike for your prokaryotes, in which the cell walls always contain a complex polysaccharide peptidoglycan. Your prokaryotes also divide by binary fission, and your eukaryotes, it's through cell division, in which there is an involvement of the process of mitosis. Now, let's talk about the cytoplasm. Again, the cytoplasm is located outside the nucleus, and it contains organelles and inclusions. Okay? And your aqueous gel is called as the cytoplasmic matrix. Your cytoplasmic matrix, which again houses your organelles and inclusion, would contain variety of solute such as your sodium, we have potassium, calcium, we also have organic molecules such as intermediate metabolites, we have carbohydrate, lipids, we also have proteins and RNA as well. So within the cytoplasm, the organelles are categorized as either membranous or non-membranous. If we say membranous, they are surrounded therefore by membranes, and that would include structures such as your mitochondria, your endoplasmic reticulum. We also have your Golgi apparatus and lysosomes. For non-membranous, therefore, on the other hand, they lack, from the word non-membranous, they lack a membrane. And it includes your ribosomes and cytoskeleton components. Now, we have here a picture in which we try to differentiate the different organelles or inclusions, their sizes, and their light microscopic and their electron microscopic features. Now, I would like us to focus on the light microscopic features. What organelles or inclusions are not visible in light microscopy? We have the following. The first one is we have the plasma membrane. Again, it's not visible in light microscope. You also have your SER or your smooth endoplasmic reticulum. It's also not visible under light microscope. We also have your endosomes. This is not also visible in light microscope. And we have ribosomes. Again, there are four organelles or inclusions that are not visibly seen in your light microscopy. You have your plasma membrane, your soft endoplasmic reticulum, your endosomes, and the ribosomes. There are also organelles or inclusions that are visible only in light microscope in um, different conditions, such as some are visible only in light microscope when stained with enzyme histochemical stain. When we talk about those types of organelles, we actually have two. We have your peroxisomes and your lysosomes. Again, your peroxisomes and lysosomes are only visible in light microscopy after a special enzyme histochemical stain um, or staining is process, or if we add histochemical stain on these types of organelles, okay, or in the specimen. And lastly, there are also organelles or inclusions that are visible only in light microscopy only when they are in large structures, and we have two. We have your cytoskeleton elements, okay, so they are only observed when organized into large structures, for example, your muscle fibrils, and you also have your secretory vesicles, in which they are observed only when vesicles are very, very large. Okay? So again, not visible in light microscopy, we have plasma membrane, SER, endosomes and ribosomes. Visible in light microscopy only when they are stained with enzyme histochemical stain. That would be your peroxisomes and your lysosomes. Visible only in light microscopy when organized into large or packed into large structures. We have your cytoskeleton and your secretory vesicles. Now let's talk about the different membranous organelles. The first one is we have your plasma membrane. Your plasma membrane is composed mainly of phospholipids. Okay, because of the fact that your plasma membrane is a lipid bilayered structure. So it's composed mainly of your phospholipids which have an amphipathic properties. Ibig sabihin, it both contains your hydrophilic heads, which is water-loving, and hydrophobic tails, which is water-fearing. We also have cholesterol, which helps in the maintenance of the membrane fluidity. Also, your plasma membrane actually contains or it has protein molecules. Namely, we have your integral protein, okay, or your integral membrane proteins, and your peripheral membrane proteins. From the word integral membrane proteins, your integral membrane proteins are therefore embedded within the lipid bilayer. And these proteins across the membrane are involved in various functions such as transport and signaling. Next is we also have your peripheral membrane proteins. And therefore, from the word periphery, okay, they are attached to the surface of the membrane. And these proteins, of course, given that they are in the surface of the membrane, are important or it plays key roles in signaling. Also, it maintains the cell shapes and other function. Again, when we talk about your plasma membrane, it primarily consists of your phospholipid, your cholesterol, and your protein molecules. Now, during cell injury, whether from disease, from trauma, or physiological stress, changes can occur in your cell's plasma membrane. And these changes could actually lead to the formation of plasma membrane blebs. What are blebs? Your plasma membrane blebs now are small bubble-like protrusions on the cell surface. And when we talk about blebbing, that is resulted from the detachment of your plasma membrane from the underlying actin filaments of the cytoskeleton. This detachment, therefore, would disrupt the membrane structural integrity and causes protrusion or bulging outward. Notably as well, cytoskeletal poisons such as your phalloidin and cytoskalasin B can cause extensive membrane blebbing. For example, for your phalloidin, this is a toxin now that would bind to your actin filaments. It will stabilize the actin filaments and it would disrupt the normal cytoskeletal function. This phalloidin can actually cause extensive membrane blebbing by preventing the proper attachment of the membrane to the cytoskeleton. We also have your cytokalasin B. Your cytokalasin B, on the other hand, this is a type of drug that would inhibit the polymerization of actin. Therefore, if it inhibits the actin polymerization, it will cause actin filaments to break down and it would lead to similar effects of extensive blebbing and cell deformity. We can see here a structure of your plasma membrane in which we would like to highlight your lipid rafts. Your lipid rafts now functions as signaling platforms in the ocean of lipids. When we say signaling platforms, it is equipped with necessary elements such as receptors, coupling factors, effector enzymes, substrates, and it would receive and convey specific signals. Okay? And in lipid drafts, usually it contains already your integral and your peripheral protein. Okay? Next is when we talk about your plasma membrane, again, it contains two non-polar chain fatty acids, which would bear your hydrophobic tail, which will face inward. Therefore, if it face inward and it's a hydrophobic, therefore it's water fearing. Okay, so it will create a non-aqueous environment that would act as a barrier, while rather the hydrophilic heads would face outward. Okay, so it will interact with the aqueous environment inside and outside the cell. Again, we also have your integral proteins and we have your peripheral proteins. I would like to highlight on integral proteins that we have your E phase and your P phase. Your E phase is your extracellular phase, and this is considered as the outer surface of the plasma membrane that faces the extracellular environment. Your P phase, on the other hand, is your protoplasmic space or your protoplasmic phase rather, and that's already the inner surface of the plasma membrane that would face the cytoplasm. So we have here a picture in which we try to observe the scarcity of molecules of your E phase, okay, as we can see, as compared with your P phase, because your E phase now, which again, when you talk about your E phase, it faces the extracellular environment. Generally, it has fewer molecules compared to the P phase. The integral proteins in your E phase are therefore less densely packed. Unlike for your P phase, okay, that again it faces the cytoplasm, it contains higher concentration of integral proteins that would project into the cytoplasmic site. And this phase, your P phase, often therefore shows a higher density of membrane associated molecules and protein structures. Okay? Now, in the plasma membrane, there are actually 6 broad categories of membrane proteins. And when we talk about your membrane proteins, these are essential for facilitating transport. Aside from that, for signal transduction, cell recognition, we also have communication, enzymatic activity, we also have structural support, cell adhesion, those different types of functions. Thereby, they maintain cellular function and interact with the environment. So as we have mentioned, there are at least 6 broad categories. We have pumps, channels, receptors, linkers, enzymes, and the structural proteins. When we talk about your pumps now actively transport specific ions and metabolic precursors across membranes using energy. Again, there is an expenditure of energy when we're talking about pumps. On the other hand, your channels would facilitate the passive diffusion of small ions, molecules, and water in either across the plasma membrane. So channels, it's through passive diffusion. Pumps, it would be uh, or it would utilize rather cellular energy. When we talk about your receptors or your receptor protein, it enables the recognition and binding of ligands to the extracellular surface of the plasma membrane. It would therefore facilitate the processes like your hormonal stimulation, coated vesicle endocytosis, and you also have your antibody reactions. On the other hand, when we're talking about your linker proteins, for example, such as your integrins, it would anchor the intracellular cytoskeleton to the extracellular matrix. So it would link now your cytoplasmic actin filaments to the extracellular matrix protein such as your fibronectin rather. Okay? We also have enzymes, and we know that enzymes would perform various biochemical roles that would include, for example, catalyzing reactions at the membrane surface. And on the other hand, when we're talking about your structural proteins, of course, your structural proteins could notably be visualized using your freeze fracture method. So it helps form junctions between the neighboring cells, and it would provide mechanical support and maintenance of the tissue structure. So those are the different categories or the 6 broad categories of your membrane proteins. Next is we also have in your plasma membrane, we have to consider that your plasma membrane is a selective barrier. It would regulate, therefore, the passage of different materials into and out of the cell. Therefore, it would maintain the constant ion content in the cytoplasm, and as well as it would carry out number of specific recognition and signaling functions. In the different mechanisms of transport across the plasma membrane, the first one is we have your passive processes. When we're talking about the passive processes, that's the movement of substances down their concentration gradient without, again, a highlight on, without using or the expenditure of your cellular energy. And in passive processes, we have two. We have your simple diffusion and we also have your facilitated diffusion. When we say simple diffusion, that's already the unassisted movement of small nonpolar substances across a selectively permeable membrane. For example, your oxygen and your carbon dioxide. On the other hand, when we say facilitated diffusion, that's the movement of ions and small polar molecules down their concentration gradient across a selectively permeable membrane via a transport protein. So there is a presence of your transport protein. For example, your glucose is transported through your GLUT proteins. Okay, so that's a family of your transport protein. When we talk about your carrier proteins or your transport proteins rather, we have your carrier proteins and your channel proteins. Okay? So if we talk about your carrier proteins, these are proteins that transfer water-soluble molecules. Okay? And they are highly selective, often transporting only one type of molecule. For example, we have your sodium-potassium pump, or you also have your hydrogen pump and your glucose carriers. So as we have mentioned earlier on, your glucose carriers, for example, it will facilitate the transport of glucose into cells through the use of GLUT transporters. For sodium potassium pump, as we know, it will actively, okay, transport sodiums or your sodium ions rather out of the cell, while it will also um, um, elicit a mechanism in which potassium ions goes into the cell. And of course, your sodium potassium pump, again, sodium outside, potassium inside. This pump actually is crucial for maintaining ion balance and cell volume. You also have your hydrogen pump. Of course, as we know, it will transport your hydrogen ions or your protons across the membrane. And this is important in processes such as your acid-base balance. You also have your channel proteins. When we talk about channel proteins, these are considered as transmembrane proteins or the integral proteins. They usually contain a pore domain that would serve as ion selectivity filter. And the transport can be regulated by membrane potentials, for example, your voltage-gated ion channels in the neurons. You also have ligand-gated ion channels that could be seen in the acetylcholine rather receptors. And you also have mechanical stress or mechanically gated ion channels such as your internal ear. Now, we can see here a picture in which we have your fat soluble and other small uncharged molecules in green, and this would represent your simple diffusion. It means that these molecules cross the plasma membrane by cell diffusion. Why? Because there is a movement or the molecules moves down to the concentration gradient without the need for transport proteins. The ones in blue naman, these are considered as your carrier proteins, okay? The small water-soluble molecules, these molecules would require highly selective proteins to facilitate their transport across the plasma membrane as they cannot diffuse through your lipid bilayer on their own, unlike for your simple diffusion. And on the red one, okay, the one in red, we could see here ions and small charged molecules. And these molecules are transported through the plasma membrane by or via ion selective channel proteins. This will allow now the passage of specific ions and small charged molecules. Typically, they go down to their electrochemical gradient. Since in channel proteins, usually it's charged. So there are positively and negatively charged molecules. Okay? And you also have your osmosis as well as part of passive processes. When we talk about osmosis, this is actually a special case of facilitated diffusion, which involves the movement of water through aquaporins. Okay? This aquaporins, what happens? It's from an area of lower solute or lower concentration to a area rather of higher solute concentration. That would be your osmosis. Next is we also have your active processes. In your active processes, there would be a movement of substances against their concentration gradient, which will therefore require the expenditure or would require the utilization of cellular energy, which is usually your adenosine triphosphate or your ATP. So in your active transport, there would be transmembrane protein pumps. So these pumps would transport ions or molecules actively across membranes. You also have vesicular transport. So that's already the movement of substances into or out of the cell via vesicles. We have here two major forms of vesicular transport. We have your endocytosis and your exocytosis. Basically, if we talk about your endocytosis, this is a process now that would internalize substances from the extracellular environment. What are the different types of endocytosis? We have phagocytosis, for example. As early as now, we should know that phagocytosis is clathrin independent uptake of large particles or cells, for example, in immune cells that would engulf your pathogens. You also have your pinocytosis. Pinocytosis as well is considered as clathrin-independent uptake of extracellular fluid and also dissolved solutes. And we also have your receptor-mediated endocytosis, which is considered as clathrin-dependent uptake of specific molecules bound to your cell surface receptors. For example, the uptake of your cholesterol in your or via LDL receptors or your low density lipoprotein receptors. That would be an example of your receptor-mediated endocytosis. You also have your exocytosis. In exocytosis, from the word exo, there is a process of expelling substances from cell via vesicles. We actually have two pathways. We have your constitutive pathway and we also have your regulated pathway. Now, in endocytosis, there is an uptake again of fluid and macromolecules. Okay? It would depend rather on different mechanisms such as your pinocytosis, phagocytosis, and receptor-mediated endocytosis. When we talk about your pinocytosis, it involves now the formation of small vesicles that would engulf the extracellular fluid and dissolve substances. Notably, in your pinocytosis, there are what we call now mechanoenzymes. And these mechanoenzymes are involved in the pinocytotic vesicle scission. This will help the separation of the vesicles from the membrane or from the plasma membrane. Your pinocytosis is considered as clathrin independent, okay? So it does not rely on clathrin dependent or clathrin coated pits for vesicle formation, differentiating it later on from your receptor mediated endocytosis, in which we consider it as clathrin dependent. And this process occurs continuously and is a regular part already of our cellular activity. So it involves in the fluid uptake and nutrient acquisition. So here we can see a picture in letter A. This is already a process of pinocytosis in which it is involved in the dynamic formation of small vesicles at the cell surfaces. And here is an electron micrograph that would show the smooth surfaces of your pinocytotic vesicles. Okay? So these are the pinocytotic vesicles. within the cytoplasm of endothelial cell of a blood vessel. Next is your phagocytosis. So in phagocytosis, there is rather ingestion of large particles such as your cell debris, your bacteria, and foreign materials. And in here, in phagocytosis, there is pseudopodia formation. When we say pseudopodia, these are plasma membrane will extend projections, okay, to engulf particles, forming now your phagosomes. And your in the process of phagocytosis, rather, it is considered as actin dependent. Okay? When we say actin dependent, it would require the dynamic rearrangement of actin filaments for pseudopodial extension. And it includes the polymerization and repolymerization of your actin. Aside from that, in phagocytosis, it's considered as receptor-mediated process. So, ibig sabihin, it often rather involves specific receptors on the cell surface that would recognize and bind particles. And as we have mentioned, your phagocytosis is considered as clathrin independent. So, it does not rely on clathrin-coated pits for vesicle formation. Now, in phagocytosis, there are what we call as specialized cells. So, for example, when we talk about specialized cells, we are talking about your macrophages. Your macrophages, this is a general term for the large phagocytic cells that is found in various tissues. Different types of macrophages are designated on different types of tissues based on their location. When your macrophages are located in the liver, it's called as your Kupffer cells. Again, it's called as your Kupffer cells if it's in the liver. We call your macrophages as mesangial cells in the kidneys. Okay? So Kupffer, liver, mesangial in the kidneys. Those macrophages that reside in your connective tissues are called as your histiocytes. The ones present in the brain tissue are called as your microglia. Those that are found in the joints are called as your synovial A cells. The ones present in the bones, for example, rather, is your osteoclast. Your macrophages located in the lungs, they are called as your alveolar macrophages. And lastly, the ones that are found in the skin are the Langerhans cells. Again, skin Langerhans cells, your lungs, alveolar macrophages. In joints, we have your synovial A cells. We also have, by the way, we also have yours in spleen, the ones located in spleen, that's your littoral cells. In the brain tissue, we have your microglia or your microglial cells. In connective tissues, your histiocytes, mesangial cells, that's in your kidneys. And again, in the liver, we have your Kupffer cells. These specialized cells are part of your MPS or your mononuclear phagocytic system, and they play very crucial roles in phagocytosis. Okay? So they play role in phagocytosis in different tissues and organs. That's why different types of macrophages based on location would have different names. So those are your specialized cells. In this picture, we could see in picture A, this is a phagocytosis of large particles such as, for example, your bacterium that has been killed as a result of immune response. We also have here in letter B, this is a non-biologic material. And usually, your non-biologic materials are attached to a non-FC receptor. Okay, again, they are attached to a non-FC receptor in the plasma membrane. What are examples of non-biologic materials? We have your carbon particles, your inorganic dust, asbestos filter, etc. Now we have here a video in which we could see chemotaxis. Okay? So in chemotaxis, what happens? Okay. As we can see in the video, this is what we call as your chemotaxis. Your chemotaxis is a directed movement of cells or an organism towards or away from a chemical source. So a classic example or a classical example of your chemotaxis is the movement of immune cells, which was presented in the video. For example, your neutrophils or the macrophages, they would move towards the chemoattractants that is released at the site of infection or injury. Some examples of your chemotaxis would include your 4-mil peptides, your chemokines, your complement proteins, okay? And when we talk about your chemokines, these are a class of cytokines with functions that would include attracting white blood cells to the sites of infection. You also have the concept of complement activation. When we say complement activation, the main function of your complement proteins is to aid in the digestion or in the destruction rather of your pathogens. They would pierce in the outer membrane, okay? If there is a piercing of the outer membrane, there is cell lysis, or it could also make them more attractive to your phagocytic cells, such as your macrophages, okay? And when we talk about this concept, you also have to talk about opsonization. When we say opsonization, that is the coating of a particle with proteins that facilitate phagocytosis of the particle. Okay? So chemotaxis and the different concepts that have been mentioned would be uh, discuss deeper later on as you would have your immunology and serology. Okay? Next is we have your receptor-mediated endocytosis. In receptor rather, or receptor-mediated endocytosis, we have your cargo receptors. Your cargo receptors are specific receptors on the cell membrane that binds to the target molecules or your cargo. This will accumulate in well-defined regions of the membrane known as your coated pits. And when we talk about clathrin, again, because your receptor-mediated rather endocytosis is clathrin dependent. This is a protein now that coats your cytoplasmic side of the budding vesicle. So it will help to form the vesicle by invaginating the membrane. Again, your clathrin, that's a type of protein that would coat your cytoplasmic side of the budding vesicle. On the other hand, we also have your adaptin. What is adaptin? That's also a coating protein complex rather that would interact with the cargo receptors and the clathrin. And your protein is instrumental in selecting and concentrating appropriate cargo molecules for internalization. Okay? So here, we can see your receptor-mediated endocytosis. Next is we also have your exocytosis. And as we have mentioned, your exocytosis would have two general pathways in which we have your constitutive pathway and your regulated secretory pathway. In the constitutive pathway, there would be a continuous delivery. So it means that the substances are continuously delivered or transported to the plasma membrane without the need of specific signaling or regulation. And this pathway, again, this pathway is present to some degree in all cells. And if we try to look it under a transmission electron microscope, your constitutive pathway would reveal that the cells utilizing this pathway typically lack secretory granules as they do not store products for regulated release. Okay? Again, if it's constitutive, it is a continuous delivery because of the fact that it does not need specific signaling or regulation. The pathway is present to some degree in all cells. And if we try to look it under a TEM, it would reveal that the cells in this pathway lack secretory granules. Examples, we have your immunoglobulins. So there is secretion. These are secretion by the plasma cells, which are produce your antibodies. You also have your procollagen. This is secretion by your fibroblasts, which will continuously release the precursor of your collagen synthesis. That's why it's called procollagen. On the other hand, in your regulated secretory pathway, there is regulation. So the secretion occurs only in response to your external signals or stimuli. This will ensure, therefore, precise timing of release. Unlike for the constitutive pathway rather, it does not need a signal or a stimuli. And in your regulated secretory pathway, we have secretory proteins. They are concentrated and they are usually temporarily stored in your secretory vesicles within the cytoplasm until there is a need of this type of pathway, or until there are appropriate signal triggers that would release your secretory proteins. Okay? And this pathway also is observed in specialized cells. And that would include your endocrine and exocrine cells. As well as also we can see it in your neurons. Okay? In endocrine cells, there is a release of hormones into the bloodstream, for example, in response to specific physiological signals. You also have your in exocrine cells, again, there is release of enzymes or other substances that through your ducts, okay, since it's exocrine, ducts to the external environments or into the internal body cavities. And as well as in your neurons, it will release neurotransmitter in response to electrical signals to communicate with other neurons or target cells. So that would be your regulated secretory pathway. And in this diagram, as we can see, it shows now the two pathways of your exocytosis. Usually, the newly synthesized proteins from your rough endoplasmic reticulum are delivered in the COP2 coated vesicles to the Golgi apparatus. Again, your newly synthesized proteins from your RER, they are delivered by your COP2 coated vesicles to the Golgi apparatus. Okay? So in the Golgi apparatus, of course, there will be modification. So it will sort, it will package the final secretory product, and it is transported to the plasma membrane in vesicles from your TGN. Your TGN stands for your trans Golgi network. Okay? So that's your TGN. The blue arrows, as we can see, it would indicate now your constitutive pathway, okay, in which your proteins leaves the cell immediately after their synthesis, okay. Again, the blue arrow would represent your constitutive pathway. On the other hand, your red arrows would indicate your regulated secretory pathway, in which secretion of protein is regulated by hormonal or neural stimuli. Okay? So, without stimuli or signals, there won't be a secretion of these proteins. So that's your constitutive secretory pathway, which is represented by the blue arrows, and we also have your regulated secretory pathway, which is represented with the red arrows. Okay? Next is we also have your endosomes. Your endosomes, we have actually both early endosomes and late endosomes. Your early endosomes are the membrane enclosed compartments involved in your endocytotic pathways. Your early endosomes are restricted to a portion of the cytoplasm near the cell membrane. So the function of your early endosome would be it would serve as the initial site for sorting and processing of internalized materials from endocytosis. Your late endosome, in the other hand, these are vesicles that are or that originate from early endosomes, and they move to the deeper structures within the cytoplasm. And typically, they mature into lysosomes. Again, your late endosomes would mature into lysosomes, where they would participate later on in the degradation of internalized materials. What would be a characteristic of your late endosome? If we try to compare it relatively to your early endosome, your late endosomes would have a more acidic pH, okay? Because it contains hydrolytic enzymes that would break down various substances, okay? So for your early endosomes, again, its localization, it's found more on the peripheral cytoplasm. The morphology, it would have tubulovesicular structure. In the state of acidification, it exhibits only a slightly acidic environment at about 6.2 to 6.5. And the function again is to sort and recycle protein internalized by your endocytotic pathways. For late endosomes, it's usually positioned near the Golgi apparatus and the nucleus. And the morphology, it's like an onion-like internal membranes. And again, it's more acidic, averaging of around 5.5 pH because of the fact that it contains hydrolytic enzymes. Here, this is a diagram now that would show the fate of your proteins. And again, your proteins is represented by the red circles. So it's endocytosed from the cell surface, and it's destined for your lysosomal destruction in the end. Okay? So what happens? Your proteins are first found rather in your endocytotic vesicles. Okay? So they are found in the endocytotic vesicles that would deliver them to your early endosomes. And again, your early endosomes are located in your peripheral part of cytoplasm. Because of the sorting capability of your early endosomes, the receptors are usually recycled. Again, the receptors are usually recycled to the plasma membrane. And the endocytosed proteins are transported via your MVB or your multivesicular bodies to your late endosomes. Again, what happens? Your receptor now is usually recycled in the plasma membrane and your proteins are usually coated or transported via your multivesicular bodies or your MVB. Going to your late endosome. Okay? And your late endosome, it's positioned near your Golgi apparatus and the nucleus. And that proteins transported to the late endosomes will eventually be degraded in your lysosomes. And as we can see here in the arrow, from 6.2 to 5.5 to 4.7, we could really see that your late endosome to your lysosome is relatively relatively have acidic pH or lower pH. And again, your MVBs, this transports your substances between your early and late endosomes, and they are considered as highly selective transporters. Now, your endosomes can be viewed either as stable cytoplasmic organelles or transient structures. When we say stable compartment or stable cytoplasmic organelle, this describes now that your early and late endosomes, specifically in your stable compartment model, this describes now that your early and late endosomes as stable. So they are distinct cellular organelles that do not change significantly over time. So each type of endosome would maintain therefore its identity and function within the cell. Okay? So that's stable compartment model. When we talk about maturation model, on the other hand, the concept of this model would be early endosomes will gradually mature into late endosomes and eventually it becomes lysosome. The transition now would involve changes in the endosomal membrane as well as your protein composition and the function. It would reflect, therefore, a dynamic process of maturation and degradation within the cell. So those are the two different models that would explain the origin and the formation of endosomal compartment. One model would mean or would actually define your endosomes as a stable compartment in which they would not change and they would maintain their identity and function. The other one is maturation. It means that from early endosomes, it will mature to late endosomes and it will become lysosome. Therefore, there are changes in structure, in the membrane, in the position, composition, and the function. And next is we're going to talk about your lysosome. In lysosome, we can see here your lysosome biogenesis. Your diagram would show the regulated and constitutive pathways for the delivery of the lysosomal specific membrane proteins into early and late endosome. Your blue arrows would indicate your constitutive secretory pathway, in which certain lysosomal membrane proteins will exit the Golgi apparatus and are delivered rather to the cell surface. So from there, they will be endocytosed and via the early and late endosome or the late endosomal compartments, they will reach your lysosome. Your green arrows would indicate, on the other hand, your endosomal Golgi-derived coated vesicle secretory pathway. Here, other lysosomal protein after sorting and packaging from your Golgi apparatus in your clathrin-coated vesicles, it will fuse with your late and your early and late endosomes. Okay? Again, in your constitutive secretory pathway, your lysosomal proteins will exit the Golgi apparatus. They are delivered to the cell surface. From there, they are endocytosed and they go to your early and late endosomes and they will reach your lysosomes. In the green arrows, which is represented in your Golgi-derived coated vesicle secretory pathway, so what happens is your lysosomal proteins, after sorting and packaging in your Golgi apparatus or your Golgi body in clathrin-coated vesicles, they will fuse with your early and late endosomes. And if we talk about your lysosomes, they are usually spherical in nature and they are present at a uniformly granular or electrodense appearance in your TEM. Your lysosomes are membrane-limited vesicles containing approximately 40 hydrolytic enzymes. And these are responsible for degrading various biomolecules. In enzyme synthesis and targeting, your lysosomal enzymes are synthesized usually in your RER or in your rough endoplasmic reticulum. And the enzymes are sorted in your Golgi apparatus, in which they are tagged with your mannose 6 phosphate or your M6P rather, M6P receptors or residues. What are your M6P receptors? These are your transmembrane glycoproteins that bind to your M6P tag enzymes, which will direct them to your lysosomes. Again, M6P or your mannose 6 phosphate receptors are types of transmembrane glycoprotein that would bind to your M6P targeted enzymes that would direct them to your lysosomes. What are common hydrolytic enzymes? The first one is we have, for example, your proteases. Your proteases would degrade proteins. We also have your nucleases. Usually, it breaks down your nucleic acids. Your phosphatases, it would remove, from the word itself, it would remove the phosphate groups from molecules. Phospholipases, it will hydrolyze your phospholipids. We also have your sulfatases or sulfatases rather. It would degrade your sulfate esters. And we also have your beta-glucuronidase. Your beta-glucuronidase rather, it will break down your glucuronic acid conjugates. So those are the most common hydrolyses. Okay? And aside from that, in lysosomes, the lysosomal membrane is resistant to the hydrolytic digestion occurring in the lumen. Okay? So it will prevent, therefore, your self-digestion. And in lysosome, it contains an unusual phospholipid structure with a high cholesterol content and a unique lipid called your lysobisphosphatidic acid. Again, it contains a phospholipid structure, an unusual one, with a high cholesterol content and a unique lipid which is called as your lysobis or your uh, phospho, phosphatidic acid. And what are the membrane proteins? For example, we have your LAMPs or your lysosome associated membrane proteins. Your LAMPs now are a group of glycoproteins that help maintain the lysosomal membrane integrity and function. So again, it's for lysosomal membrane integrity and function. We also have your LMPs, okay? Or your lysosomal membrane glycoproteins or LGPs rather. Your LGPs are glycoproteins that are involved in various lysosomal processes that would include now your enzyme transport and your membrane stability. Next is your LIMPs or your lysosomal integral membrane proteins. Your LIMPs naman, they are embedded in the lysosomal membrane and they contribute to its unique properties and functions. So those are the different membrane proteins. Again, your LAMPs rather, that's for integrity and function. Your LGPs, that would be for your enzyme transport and membrane stability. Your LIMPs, this is actually for, or these are embedded in the lysosomal membrane and they contribute to the properties and functions as well. So here we can see a diagram in which your lysosomal membrane proteins are actually synthesized in your RER and they would have specific lysosomal targeting signal. And also your lysosomes and your late endosomes contain hydrogen ions or proton pumps and transport proteins that would transport your final products of digestion. Okay, we have here a clinical significance in which we're going to talk about your chloroquine. Your chloroquine now is a type of lysosomotropic agent that is used for the treatment and prevention of malaria. Okay, so what happens here? What would be your mechanism of action of your chloroquine? Your chloroquine will accumulate in the lysosomes due to its lysosomotropic properties. Again, because if we talk about your chloroquine, it's a lysosomotropic agent. So what happens? It raises the pH with your or within your lysosomal lumen. Okay? If it raises pH within your lysosomal lumen, it will therefore inactivate many lysosomal enzymes. And what would be the effect therefore in malarial parasites such as your Plasmodium falciparum? Your chloroquine will concentrate in the acidic food vacuole of your malarial parasite, which will disrupt therefore its digestive processes. Okay? So your chloroquine therefore, by raising the pH and by interfering with the lysosomal enzyme, because it will inactivate these enzymes, your chloroquine would therefore prevent the parasite, the malarial parasite, from digesting hemoglobin and other necessary substrate. And therefore, this disruption of your chloroquine will ultimately lead to the death of the parasite, which would make your chloroquine an effective treatment and prevention for malaria. Okay? Next is, we have to consider that your lysosomes have three pathways of delivery. We have extracellular large particles, you also have your extracellular small particles, and you also have intracellular particles. Okay? In here, we could see that through phagocytosis, it will engulf now your large extracellular particles, such as, for example, your bacteria, and in pinocytosis and your receptor-mediated endocytosis, it will internalize small extracellular particles and molecules. And also in autophagy, it will isolate and it will degrade intracellular components by enclosing them in the autophagosomes that would fuse with your lysosomes. So here, we could see the pathways of delivery of materials for digestion in the lysosomes. In the endocytic pathway rather, which is in red, this pathway, of course, is responsible for the degradation of your extracellular substances. So we have your nutrients, your membrane proteins, and your ligands, for example. For the phagocytotic pathway, this pathway is crucial again for the immune responses. So it will also be responsible in the clearance of your dead cells and pathogens. And in your autophagic pathway, that would be a pathway now that plays a key role in cellular maintenance. So it will recycle cellular components if necessary, and it is used in the response of nutrient deprivation. Okay? And also, in your lysosomes, there are three well-characterized pathways of autophagy. We have your macroautophagy, microautophagy, and your chaperone-mediated autophagy. In your macroautophagy, this is a non-specific process. So in here, there would be a formation of a autophagosome. So what happens? A portion of your cytoplasm or an entire organelle, for example, would be engulfed by a membrane derived from the endoplasmic reticulum, which is known as your isolation membrane. This will form a double membrane structure, which we call now as your autophagosome. And as we talk about maturation, the autophagosome will mature by fusion or by fusing it with a lysosome. And therefore, it would result to a formation of autolysosome. From autophagosome plus your lysosome, its fusion will create your autolysosome. And later on, inside the autolysosome, the isolation membrane will disintegrate and the contents of are exposed, 'di ba, in the hydrolytic or through the hydrolytic enzymes. And this process would lead to the degradation of the enclosed cytoplasmic materials, which will include, for example, damaged organelles, we have aggregated proteins, and other types of cellular debris. Therefore, if we talk about the function of your macroautophagy, this is now important in maintaining cellular homeostasis because it removes damaged or unnecessary components. Therefore, it will support the function and the health of the cell. Next is we also have your microautophagy, which is also considered as a non-specific process. So in here, a small soluble cytoplasmic proteins will be internalized by your lysosomal membrane through a process of invagination. So the invagination of the lysosomal membrane will create your small vesicles. That's why it's called microautophagy. It will create small vesicle that enclose the cytoplasmic proteins. And these vesicles would fuse later on with the lysosomal lumen where the enclosed proteins will be degraded by your lysosomal enzymes. Therefore, the function of your microautophagy would ensure that there is a continuous removal of small cytoplasmic proteins. Thereby, it will help in the maintenance of your homeostasis because it will manage now the turnover of soluble proteins under normal physiologic conditions. Next is we also have your chaperone-mediated autophagy. And your chaperone-mediated or chaperone assistance is considered as a selective process. It will require specific cytosolic chaperones, such as, for example, your heat shock chaperone protein HSC73, which will assist in the recognition and transport of substrate. So this process is activated during nutrient deprivation. Therefore, the function of chaperone-mediated autophagy is it is responsible now in the degradation of approximately 30% of cytoplasmic proteins in organs like your liver and kidneys. Okay? And in here, we could see your macro-autophagy, your micro-autophagy, and your chaperone-mediated autophagy. Again, in your macro-autophagy, this is in which we could see it depicts now that a portion of the cytoplasm, or again an entire organelle, is surrounded by an intracellular membrane of the endoplasmic reticulum, which will form the double membrane autophagosome. After fusion with your lysosome, the inner membrane and the content of the vacuole are degraded. In your microautophagy, your cytoplasmic proteins are internalized into the lysosomes by invagination of the lysosomal membrane. And again, in your chaperone-mediated autophagy, the chaperone protein, that is your HSC73, would bind to the protein and it will help transport it into the lysosomal lumen where it will be degraded. Okay? So in lysosomes also, we have your heterolysosomes and your residual body. Your heterolysosomes are active lysosomal organelles which is involved in the digestion of variety of materials. So if we look into the appearance of your heterolysosomes under your transmission electron microscopy, they are generally larger and they have more heterogeneous appearance. Okay? This is due to the fact that your heterolysosomes have diverse types of materials that they process. Okay? That's why they are generally larger and more heterogeneous when we look into it under a transmission electron microscope. And again, the function of your heterolysosome that would digest various substances that would enter the lysosome from different cellular processes such as, for example, through your endocytosis and your phagocytosis. We also have your residual body. Your residual body is a vacuole now that is filled with undigested debris that would result from the incomplete hydrolytic breakdown of some of your lysosomal contents. What would be the characteristics of your residual body? First one is cell aging. So there is accumulation of residual bodies, okay, or the accumulation rather of your residual bodies is a normal feature of cell aging. And also, your residual bodies can accumulate as lipofuscin. Your lipofuscin now is a brownish pigment that is associated with aging and cellular damage. And if we talk about the function of your residual body, definitely it will reflect now the long-term storage of your undigested materials, and it may also indicate the inefficiency already of your lysosomal degradation processes, in which we try to correlate it in your lysosomal storage diseases. Your lysosomal storage diseases are genetic disorders caused by pathological accumulation of undigested substrate in your residual bodies. So these conditions usually arise from the absence or deficiency of specific lysosomal enzymes that is required for the breakdown of various substrate. Okay? So what are the mechanisms of these diseases? For example, first mechanism rather is we have enzyme deficiency as stated. So the lack of essential enzyme will prevent the complete digestion of the substrate, and it would lead to accumulation within their lysosomes or within the lysosomes. We also have substrate accumulation. So the undigested substrate would accumulate in the residual bodies. This will cause, therefore, cellular dysfunction, and it would contribute in the aggravation of the different symptoms of these diseases. And these are the summary of the common lysosomal storage diseases. These are examples now of your LSDs, for example, your Tay-Sachs disease, okay, in which the protein deficiency would be your beta hexosaminidase or your alpha subunit. You also have your Gaucher disease, in which the deficiency would be your enzyme glucocerebrosidase. You also have your Fabry disease and other types of LSDs, okay, that is associated with the accumulation of different types of substrates due to the specific enzyme deficiencies. Next is we have your proteasome mediated degradation. Proteasome mediated degradation is a cellular process for destroying abnormal or regulatory proteins. So your proteasomes now target misfolded, aside from misfolded, denatured, or proteins with abnormal amino acids that cannot properly process and function. Okay? The regulation of your proteasomes, they degrade normal short-lived regulatory proteins that needs rapid inactivation. For example, your mitotic cyclins. Your mitotic cyclins would regulate your cell cycle progression. Your transcription factors also is involved in regulating gene expression. Your tumor suppressors and your tumor promoters, they modulate your pathways related to your cancers. So those are the different um, those are the different things that your proteasome mediated degradation do. And your proteasomes now are large cytoplasmic or nuclear protein complexes. And they have the function to break down proteins into smaller peptides and amino acids which are recycled and further degraded. So, in comparison with your lysosomal pathway, your proteasome pathway would operate independently of your lysosomes, and it's crucial for managing proteins that needs to be quickly removed or regulated. Unlike for your lysosomal pathway, in which your lysosomal pathway, from the word itself, involves the lysosomes for degrading the larger cellular components, and that would include your organelles and extracellular materials. Here we have your polyubiquitin change, or at least for ubiquitin molecules. This change tag the target protein and they will serve as degradation signal for the 26S proteasome complex. Your 20S core proteins that would facilitate the multi-catalytic protease activity, in which your polyubiquitinated protein is degraded into small polypeptide and amino acid. Your 19S RPs, or regulated or regulatory particles rather, on the other hand, would unfold the protein and would lead to its entry to destruction chamber. And your 19S regulatory particles, on the other hand, on the base, okay? So again, the 19S regulatory particle on the lead that would unfold the protein and lead to the entry of to the destruction chamber. While in your 19S regulatory particles, in your base, that would release the short peptides, okay, or the short polypeptides and amino acids after completion of degradation. Now, there are different clinical significance. So if there is a malfunction of proteasome-mediated degradation, we have this following diseases. We have your Angelman syndrome. In Angelman syndrome, there are mutations in your UBE3A. Okay, again, there are mutations in your UBE3A that would cause inhibition in the proteolytic activity of your proteasomes or your proteasomes. Your Alzheimer's disease, there is an accumulation here naman of your amyloid and your tau hyperphosphorylation. It will impair your proteasome function. And in HPV or your infections with your human papilloma virus, the virus would use your ubiquitin proteasome system or your UPS to complete your various life cycle. Okay? So those are the different diseases. Now we go to your rough endoplasmic reticulum. Your endoplasmic reticulum, or in general, it would play a crucial role in the synthesis of proteins and lipids. Aside from that, it can also serve as pathway for the transport of materials within the cell from the nuclear envelope to the membrane. So the function is for synthesis and for transport. And your it encloses a series of intercommunicating channels and sacs which we call as your cisternae. And within your TEM, the RER appears as series of interconnected membrane limited flattened sacs which we call as again your cisternae with particles studding the exterior surface of the membrane. Your RER consists of sac-like and parallel stacks of flattened cisternae which we have seen in the picture, and your ribosomes are usually studded. Okay, so your RER is studded, or it is attached with your polyribosomes, which gives now your rough appearance. That's why we call it as your rough ER. What are the different functions of your RER? First is for the synthesis of protein and also for segregation. So it synthesizes your proteins that are not destined for the cytosol, and including those meant for the secretion or incorporation of or into your cellular membranes. Also, it begins the process of glycosylation for glycoproteins. It performs modifications on newly formed polypeptides that would include folding and assembly into multi-chain proteins. And also, it is abundant in cells specialized for high levels of protein synthesis. Okay? So that's your functions. Again, it's for protein synthesis and segregation, initial glycosylation, post-translational modifications, and also assembly of multi-chain proteins. Next is, what are the components of your RER? The RER, with its ribosome-studded surface, is central to your protein synthesis. Your ribosomes, again, is attached to your RER, and the ribosomes are the sites of your protein synthesis, translating your mRNA into polypeptide chains. What are the cellular characteristics? So first one is we have basophilia. The cells engaged in high levels of protein synthesis would exhibit basophilia due to the abundance of RNA, which is prominent in your ribosomes. Also, in secretory cells, the rough ER appears as ergastoplasm in light microscopic images. This term refers to the basophilic cytoplasmic area indicative of the active protein synthesis. So in your RER, it is where the protein synthesis processes, transcription, and translation occurs. So in transcription, it takes place in your nucleus. So the process is the genetic code for a protein is transcribed from a DNA to pre-RNA, and from pre-RNA, it undergoes post-transcriptional modifications to become mRNA. And in translation, it occurs in your cytoplasm, primarily in your ribosomes. And the coded message in your mRNA is read by your ribosomal complexes to a polypeptide chain and formed by the binding of a single mRNA molecule to multiple ribosomes, which enables now efficient protein synthesis. Now, there are few diseases that are characterized by an inability of the RER to export a mutated protein to the Golgi apparatus. We have, for example, your alpha-1 antitrypsin deficiency. Your alpha-1 antitrypsin deficiency or your is a genetic mutation that leads to a single amino acid substitution in your alpha-1 antitrypsin protein. Again, it's a genetic mutation that leads to a single amino acid substitution in your A1AT or your alpha-1 antitrypsin protein. So what would be the effect on the RER? The mutated A1AT is unable to export or it's unable to be exported rather from your RER to the Golgi apparatus. And this results now to the accumulation of your defective or mutated A1AT within the RER of your liver hepatocytes. What would be the consequences? In the liver, the abnormal deposition of your A1AT in your liver cells would lead to impaired function of the liver, and it could actually cause potential liver damage. Aside from that, in your lungs, the decreased levels of functional A180 because there is mutation, of course, it would lead to unregulated activity of your neutrophil elastase. So again, normally your A180 will protect the lungs by inhibiting your neutrophil elastase, and your neutrophil elastase, that's an enzyme that would digest damaged cells and bacteria, which will prevent it from attacking healthy lung tissue. And the outcome, because of inadequate A180 that allows elastase to, it will allow now your neutrophil elastase to damage the lung tissue, which will contribute to conditions such as emphysema, which is a chronic obstructive pulmonary disease, and also, again, impaired liver function. Now, the differences between your ribosomal structure, for example, in your prokaryotic ribosomes or in bacterial, so the size now is 70S ribosome. So it consists of a 50S large subunit and a 30S small subunit. And what would be the unique feature? Your different ribosomal RNA or your rRNA and the protein composition compared to your eukaryotic ribosomes. In your eukaryotic ribosomes, it's usually 80S ribosomes. This is composed now of your 60S large subunits and your 40S small subunits. And it's slightly different from rRNA and protein structure compared to your prokaryotic ribosomes. Okay? Now, researchers have developed antibiotics that specifically targets bacterial ribosomes without affecting the eukaryotic ribosomes. What would be the mechanism? These antibiotics bind to distinct portions of bacterial ribosomes which will inhibit the protein synthesis and will effectively treat bacterial infections while minimizing harm to the host cell. So examples of those antibiotics would be your aminoglycosides, for example, your streptomycin, macrolides, your erythromycin, lincosamides, which is your clindamycin, tetracycline, which is your tetracycline, your chloramphenicol, which is also your chloramphenicol. Okay? And now let's go to your ribosomes. Your ribosomes synthesizes proteins that are destined for secretion out of the cell or for incorporation in the cell membrane. These proteins now are transported to the Golgi apparatus for further processing, packaging, and secretion. Again, that's the primary function of your ribosome for the protein synthesis, or it is the site of protein synthesis. The ribosomes in cytosol, usually, the composition is it composed of your 4 segments of rRNA and also approximately 80 different proteins. The structure, the subunits, it's composed of 2 subunits of different size, and its function is to catalyze the process of protein translation by binding to your mRNA. On the other hand, your polyribosomes, which is also known as your polysomes, it's a multiple ribosomes that is attached to a single mRNA. And the function, it simultaneously translate your mRNA into protein, which will increase the efficiency of protein synthesis. And in the appearance of your polyribosomes or your polysomes, it's intensely basophilic. Why is it intensely basophilic? Because is due to the high concentration of your rRNA and ribosomal proteins. Now, going to your smooth endoplasmic reticulum. The appearance of your SER is it lacks your bound polyribosomes, resulting in a non-basophilic staining pattern. So there is a continuity. It's continuous with your RER, and the cisternae is often more tubular and appear as interconnected channels. The functions of your SER, it's involved in the breakdown of and synthesis as well of your glycogen and lipids. It facilitates the detoxification of harmful substances. It stores and releases calcium ions as needed, and also it synthesizes phospholipids necessary for membrane formation. Now, your smooth ER houses a variety of enzymes tailored to its diverse functions. These enzymes includes, for example, your hydrolases. Your hydrolases, these are usually enzymes that would catalyze hydrolysis reactions. So there is a breaking down of larger molecules. We also have your methylases. These are enzymes involved in methylation process. We have your G6P or your glucose 6 phosphatase. These are enzymes now that plays a key role in glucose metabolism, which will convert your glucose 6 phosphate to your glucose. We also have your ATPs. These are enzymes that will hydrolyze your ATP to ADP and inorganic phosphate, which is often involved in energy transfer. And we also have your lipid oxidases. Lipid oxidases, on the other hand, are enzymes rather that would facilitate your oxidation of lipids. So it will contribute to lipid metabolism and detoxification processes. So here, this is an electron micrograph of SER. Next is we have your Golgi apparatus. Your Golgi apparatus is named after Camillo Golgi or Camillo Golgi, who discovered this organelle in 1898. What are the functions of your Golgi apparatus? It will further modify your proteins after their synthesis after their synthesis in your endoplasmic reticulum. Also, it will sort and package proteins for transport to their correct destinations within or outside the cell. Okay? So, your Golgi apparatus, we have your transport vesicles. Your transport vesicles, these are small membrane enclosed carriers that would transport your RER to the Golgi apparatus. Your Golgi apparatus have 2 phases, and that would be your cis phase and your trans phase. Your cis phase is the receiving region where your transport vesicle or your vesicles rather merge with your Golgi. Your trans phase, on the other hand, would be the shipping region where larger saccules or vacuoles will carry completed products to their destination away from the Golgi. And also, if we're talking about post-translational modifications, proteins and lipids would undergo a series of modifications as they pass through your Golgi stacks. Okay, so this includes the remodeling of your N-linked oligosaccharides that were initially added in your RER. Next is we have your mitochondria. So as we know, your mitochondria is the powerhouse of the cell. What I would like to highlight in your mitochondria is we have this hypothesis or this theory which we call as your endosymbiont theory. Your endosymbiont theory proposes that certain organelles in eukaryotic cells, specifically your mitochondria and also we have your chloroplast, originated from free-living prokaryotic organisms that were engulfed by a primitive eukaryotic cell. So, it is believed rather that your mitochondria have originated from a proteobacteria, for example. And while your chloroplast is originated or it's thought to have originated from cyanobacteria. Again, mitochondria, proteobacteria, your chloroplast, your cyanobacteria. So the engulfed prokaryotes now form a symbiotic relationship with the host cell. And it will provide advantages such as, for example, for your mitochondria, ATP production, or for your chloroplast, it's through photosynthesis. What are the evidences that this hypothesis or theory could be possible? Number one is the double membranes. Both your mitochondria and your chloroplast actually have double membranes, which is consistent with engulfment. Aside from that, there is presence of genetic material. These organelles actually contain their own circular DNA, which is quite similar with your bacterial DNA. At the same time, they have their own ribosomes, which is similar again to bacterial ribosomes than to your eukaryotic ribosomes. Aside from that, reproduction. Your mitochondria and your chloroplast will reproduce independently of the cell through the process of binary fission. And at the same time, therefore, with those evidences provided, the endosymbiont theory or hypothesis would explain the origin of your eukaryotic cells and the presence of organelles with the prokaryotic characteristics within them. Okay? Now, your mitochondria is present in cells or in all types of cells except for your RBCs and your terminal keratinocytes. And then when they are present in large number, it can cause acidophilia of your cytoplasm because of the large amount of membrane they contain. Also, we have here, again, your mitochondria would possess two membranes. We have your inner rather mitochondrial membrane, which surrounds a space which we call rather as matrix. And we also have your outer mitochondrial membrane, which is in close contact with your cytoplasm. Here we could see the structure of mitochondrion in an electron micrograph. Next is we have your peroxisomes. Your peroxisomes are spherical granules enclosed by a single membrane. So it's similar in the structure to lysosomes, but they do not contain lysosomal hydrolysis. So in here, there are different types of functions. First is your oxidases. So oxidases is an enzyme that would remove your hydrogen atoms from various substances and transfer them to your molecular oxygen or your O2, which will produce your H2O2 or your hydrogen peroxide. Aside from that, we also have your catalase. Your catalase will break down your hydrogen peroxide into water or potentially into water, which will, and also in your oxygen where it could potentially or thereby it will prevent damage to your cell. And the significance of your peroxisome, it plays actually a crucial role in cellular metabolism by participating rather in the oxidative reactions and detoxifying harmful substances because it has your oxidases and catalases. And your peroxisomes have two forms, okay? So or your peroxisomes rather forms in two ways: through budding of the precursor vesicles from the ER or growth and division of pre-existing peroxisomes. Now let's talk about your non-membranous organelles. The first one is your secretory granules. Your secretory granules are membrane-enclosed organelles which are found in cells that store products until they are released by exocytosis. These granules are concentrated. It means that they contain a high concentration of secretory products, and the release of their contents is signaled by metabolic, or if not metabolic, by hormonal or neural messages. We also have your zymogen granules. Your zymogen granules, these granules contain a dense concentration of digestive enzymes in active form. So we have your zymogen. And the function is they are found in cells such as your pancreatic acinar cells, and they are involved in the storage and release of digestive enzymes. Next is we also have your microtubules. Your microtubules are fine cylindrical structures organized in large arrays called as your axonyms in cilia and your flagella. And it's composed of a alpha tubulin and beta tubulin subunits arranged in a helical pattern. So what are the functions? It will provide, number one, it will provide structural support to maintain the cell shape. And it can also rapidly disassemble or reassemble, which will allow cellular flexibility in and movement. And what could be its role in your cilia and flagella, for example, or what could be the role rather of your cilia and flagella? It will form the core structural component of your axonym, which is essential for the movement of these organelles. We also have your microtubules. These are the different functions of your microtubules. First, it can facilitate the movement of vesicles and organelles within the cell. Aside from that, it will form the structural basis of cilia and flagella, which enable them to move, or which enables their motility. It also assists in the attaching of chromosomes to the mitotic spindle during the cell division, which will ensure proper chromosome segregation. It will contribute as well to cell elongation and motility by providing structural support and tracks for cellular movement. And also, it helps in your maintenance and regulation of the cell shape by providing rigidity and structural support. We also have your microfilaments. Your microfilaments are important to support the attachment and movement of membrane proteins, which will facilitate various cellular functions. Aside from that, it will provide structural support to microvilli, which will enhance the area of absorption. It could also enable cell movement through processes like amoeboid movement and muscle contraction, and it could contribute to the extension and formation of cell processes such as philopodia and lamellipodia, which is important for cell signaling and migration. We also have your intermediate filaments. Your intermediate filaments, it will provide structural support to the cell, and it helps maintain the shape and integrity of the cell. It can also contribute to the cell's mechanical strength by anchoring the organelles and other cytoskeletal components in place. And at the same time, it could also participate in the formation of your desmosomes. Aside from desmosomes, you also have your hemidesmosomes, which will link the cells to each other and to other extracellular matrix. We also have your intermediate filaments, in which we have your five major classes. We have your keratin. Usually, your keratin is present in your epithelial cell and it provides structural support and resilience in your epithelial tissues. Your vimentin, it will support the cellular structures and integrity, which is often actually your vimetin rather is um, often used as a research rather as a marker. Your neurofilament, it will provide structural support to your neurons and help maintain axonal diameter. Your lamins also can support the nuclear envelope and organize chromatin within the nucleus. We also have your desmin. It will provide structural support to muscle cells and link contractile elements. And we also have your glial filaments. Your glial filaments supports and the structural integrity of your glial cells in the nervous system. So we also have different cytoplasmic inclusions. For example, your fat droplets, or we can also call it as your lipid inclusions. It's nutritive inclusions for cell metabolism. Glycogen also, these are highly branched polymer and a storage material for glucose. We have lipofuscin. This is what we call now as the wear and tear pigment, and as we have mentioned earlier on, this is actually brownish gold in HNE stain. And we also have your hemosiderin, which is your iron storage complex within the cytoplasm. Okay? And usually your hemosiderin is formed by the indigestible residues of your hemoglobin. So it's present in the phagocytosis of RBCs. We also have your centrosome. This acts now as center for activity of your which is associated with your cell division. It can also organize your microtubules that will serve as an MTOC or microtubule organizing center, and also it plays a crucial role in organizing the mitotic spindle during cell division. And its appearance in electron microscopy, it would appear as a hollow cylinder closed at one end and open at the other. Okay? So it's like a tube, cylinder tube. Your centrioles, it's of course prominent in mitosis, where they will help organize the mitotic spindle and it will ensure accurate chromosome segregation. Also, it's essential for the formation of cilia and flagella. So it will serve as basal bodies. Also, it acts as sites for the assembly of your epithelial cell. And typically, the structure of your centrioles, it will appear as cylindrical structure which is composed of 9 triplets of microtubules arranged in a pinwheel pattern. Again, it's arranged in a pinwheel pattern. Next is we have your flagella, of course, which is a long, semi-rigid helical hollow tubular structure which is mainly composed of your protein, which is your flagellin. And we also have your cilia, which are hair-like processes. So, very numerous siya in epithelial cells in your upper respiratory tract, parts of female and reproductive tracts, and your ependymal lining now the cavities of your central nervous system. So, those are the different parts of your cell. By the next video, we're going to discuss the nucleus.